So I came at it as an undergrad. I was sitting in a room with people. We were talking about the things that mattered, like what is time and what is consciousness and what happens if you have a brain. And what I noticed over and over was this almost deference to neuroscientists when it came to some of the details about how the brain works when we were doing philosophy of mind conversations.
Mm.
And I was young enough to be kind of to succumb to this. And
Mm-hmm.
so I ended up just kind of realizing, okay, well, yes, we could talk about brains in a vats, but down the road in a lab, people are taking brains out of previously living animals, bathing them in fluids of salt and enough nutrient to keep them alive for a couple of hours or days, and people are studying this. And when we were having conversations about free will and what is identity, what does it mean to be the same person over time, what does it mean to choose your own preferences, can you choose your own preferences, all these things, I thought I had just read some, I think, Discover magazine article about Toxoplasma by Carl Zimmer, and that started it all. I was like, well, why don't we just study this instead of talking about it like this is a thing, this is in the sphere of empirical studies, we can look at this, we can do this. And so I kind of did it exclusively and entirely to try to study free will from some kind of oblique angle.
That was the voice of Patrick House. He's a neuroscientist and author, and he spent a lot of time researching the parasite Toxoplasma Gondii. This is a parasite that can infect all mammals, but it can only reproduce in the intestines of a cat. So in every case, it needs to make its way back to a feline to complete its life cycle. So Toxoplasma most commonly infects rats because they often live in such close proximity to cats. And it affects the behavior of the rats, causing them to lose their fear of cats and actually feel drawn to them. This is accomplished by a neurological mechanism that scientists still don't completely understand, but we know that Toxoplasma creates hundreds of cysts in the brain of its host, causing dopamine levels to rise. And dopamine is a neurotransmitter that plays a role in mediating powerful emotions, such as desire and fear, which helps to explain much of the behavior we see in mammals infected with the parasite. You heard Patrick reference identity and free will in that opening clip, where he was recounting how he first got interested in this subject. And those are the intuitions I really want to get at here. Because if a tiny parasitic protozoan can tunnel its way into your brain and stealthily alter your feelings and behavior, where does that leave room for the reality of stable identity and of self? Since it's all brain processing to begin with, it becomes hard to see where you can draw a line between things like parasites and the chemicals they release, and all of the different neurotransmitters and hormones and countless brain processes that normally govern our feelings, thoughts, and behaviors in every moment.
People had looked at Toxoplasma for about 100 years. It's been quite relevant to livestock and cattle. It's been, and it was an epidemiological problem for a very long time. It was one of the leading causes of HIV-related mortality in the late 80s. The guy at the end of Train Spotting dies of toxoplasmosis. It's neurologically, it has been known around and studied for decades and decades. But it wasn't until the kind of mid 90s or early 90s that someone, a kind of behavioral ecologist, neuroscientist out of Oxford, asked, like, well, what is it doing in these rodents? What is it doing in a mouse brain? What is it doing in a rat brain? Why is it there? And at some point, people had kind of pieced together that its definitive host is the cat. So maybe I see it as almost like a mash of cultural themes and scientific themes where people had just started to kind of uncover some of these parasite host manipulation frameworks. In media, people have been writing about, filmmakers have been directing movies where characters are imbued with something that makes them behave differently. All of these are just in the general category of unobservable change in human behavior, and the plot, the theme, the interest is in like unpacking what and why. And so it's a remarkable coincidence that the most studied animal model happens to also be the host of this thing, the indirect host.
Right.
So the mouse, like, if it was some strange exotic bird that was the intermediate species that would have been much, much harder to study. But
Yeah. Yeah.
what a world of coincidence
We
where
already have
you
these
could
in the
actually
lab.
study. Yeah, you could. And so to me, that was actually part of the beauty of the
Mm,
scientific project was most neuroscience, when you're dealing with animal models or laboratory stuff, it's always some sort of proxy between the thing you're studying and what you really care about, which is human cognition and human consciousness. I mean, that's mostly what most people care about. And in most of those cases, no matter what you do, no matter how well you solve a unanswered question in a mouse brain, you're still making an inferential leap to leap up to the human brain. And what I loved about studying toxoplasma was that the human is the model. The phenomenon of interest is happening in the mouse brain. And when we get it, we are the ancillary hosts. We are the animal model. And so I loved actually studying the thing in and of itself without having to make, with making the fewest logical leaps that I had to.
yeah. We're juxtaposing what we are kind of categorizing as the normal brain of a rat or of a human, and then the influence of this external thing on the behavior, which I think is in itself fascinating. But for me, my interest in it really came from trying to understand how much of that sort of process is already occurring in all behavior. How much is everything from the structure of the brain to any collaborations it finds itself in with parasites or diseases or injury or just normal development. Can we kind of view our behavior in the same way, and so that it shakes up our intuitions about what we normally view as inherent to ourselves, and we have this intuition that we have something that is like a soul or a self. And my interest in parasites in general, but in all of the things that I cover in my book, and my interest in neuroscience really is kind of understanding to what extent we are just experiencing processing, and to what extent free will is truly an illusion. And I'm curious how much that type of curiosity was driving you, and if it influenced your thinking on free will and other areas of consciousness.
Yeah, so I got into it for a few reasons. One of which is a little bit pedestrian, and is just really about the kind of marvel of the anomaly of the parasite host relationship. So, just to get this out of the way, what I found so fascinating was that there are all kinds of other examples for this kind of host behavior manipulation, but in general, they tend to happen in insects. They tend to happen. So there's a thing, it makes ants go somewhere, that makes crickets, all these things. And they get brought up in the same category. But I think that's a category error. And the reason I think toxoplasma is so, so, so interesting is that it gets into mammalian brains. So usually things that get into mammalian brains kill the host immediately. It is the Fort Knox, the blood-brain barrier. The mammalian brain is exquisitely protected. It's immunoprivileged. It has very few cells that can come and go. It is extremely well guarded. The fact that this thing gets in there, to me, just on a just pure almost engineering level, I wanted to know the engineering of how it does that. And it's so successful. It's in a third of human brains. It's in most livestock. It's incredibly successful. It, like malaria, so it's intimately related to malaria.
Hmm.
They're cousins or
I
third
didn't know
cousins or
that.
something.
Okay. Uh-huh.
So that protozoan, Plasmodium falciparum, and Toxoplasma, also a protozoan, they're the same kind of life form.
Hmm.
And they're just so good at what they do over and over. These are kind of unbeatable pathogens. And when a kind of indirect host ends up ingesting it, so it's almost always through, you have to eat it. So even just there, we have a vat of xenomorph level hydrochloric acid sitting in our stomach to kill supposedly everything that we encounter, right? Like we're supposed to be able to eat raw meat on the African savannah.
Right.
And so this thing avoids the vat of hydrochloric acid that we have, is fine, just probably thrives, does the backstroke, and ends up inside the immune cell that is supposed to actually kill it. So the things that are most damaging, HIV, toxo, these, I mean, broadly speaking, they use the immune system to their advantage. They Trojan horse their way in. And so Toxo does, just statistics and Brownian motion, end up in some of these immune cells which have privileged access to the brain. So a few do, just in case something gets in there. And it actually kind of wiggles inside of one of those. Instead of being killed, it actually hides inside, straight Trojan horse style. And then when it's in the brain, just pops out and lives forever and incurably
Mm-hmm.
there as a cyst in the host. And what's so remarkable to me about that identity question and the free will question, and what really kind of switched my interest into a passion regarding toxoplasma, was that it actually sits inside of neurons.
Mm.
So when these cysts form, the outside of a neuronal cell body is scary because that's where the immune systems patrol and all those immune cells patrol. But inside of a neuron is safe harbor. And so the fact that this thing goes all the way from just like the outside world, Trojan horses its way into the brain, and then finds itself, you know how we've kind of domesticated the things that scare us. We took bears and carved them into bearskin coats, and we took wolves and turned them into purse-sized little puppies and all these things. Toxoplasma, like, it effectively hides itself in a neuronal skin coat, the same way we used to hide ourselves in bearskin coats. And I just found this kind of so pleasant to think about on an
Yeah.
evolutionary level that
Yeah.
it figured these things out.
No, I
But
I get that.
and
Yeah.
in terms of identity and the free will question, like ignoring all of the process and the way it gets into the brain, let's just, cross that out and just say, okay, it's in a brain, it's in a mammalian brain, it's inside of neurons. Does that animal or humans decision making, do we call it the person? So when a person makes a decision and they have toxoplasma, are they them making the decision? Are they them plus the parasite?
Mm-hmm.
Like, how do you kind of carve out these borders between self and non-self when the thing is inside your damn neuron?
Right.
Like, if you were, I imagine, like a coloring book, and you tell someone, okay, here's an MR scan of your brain. We've abstracted it out to some bold lines so you can color the parts. Now here, color the parts that are you and not
That are
you.
you.
Are they just going to
Yeah.
make little are they going to make little holes for the thousands of parasites that are in their brain and say, like,
Right.
oh, well, everything else around it is me, but that little thing is not me?
And even what does me mean in that sentence? What are we even trying to define?
Absolutely. That it skews preferences was another thing that I found so
Mm-hmm.
interesting. So
Mm-hmm.
there was some
Mm-hmm.
talk in the early 2000s about the holy grail of a visual illusion would be if you could make the same stimuli appear black,
Mm-hmm.
pure Pantone black, and pure Pantone white. And if you could make the same stimuli, just changing the outside context and conditions from black to white, then you have achieved the holy grail of visual illusion. It cannot get better than that because you've gone from both extremes.
Mm-hmm.
And when I heard that the parasite toxoplasma for a mouse or for a rat changes a fear, an innate fear, into what might be an attraction, to me, that's the black to white. That's the holy grail of behavioral manipulation. If you can take something that you're supposed to be so afraid of and turn that into the entire opposite behavioral outcome, then everything else is in the middle.
Yeah.
Every other emotion, preference, behavior is just in the middle of that. And so
Yeah.
that
No,
that
that's
really
why it's such a
fascinated me
great
about this problem.
example. Yeah. Did you end up doing research with human subjects as well, or were you, even if you haven't, the only research I'm aware of is, I think, from, Natalie Angier did some reporting on it in the New York Times, and maybe a couple of other mainstream science articles. But I'm curious about everything I don't know about studies that were done in humans.
I never did work in humans. Again, I found the purity of the human being the animal model in this situation. The mice are clones of each other and still have individual variation, and then to deal with, the mice are 100 years inbred, clonal as you can get, and yet still there's massive individual variation in their behavior and their response to toxoplasma. So the idea of introducing the human level of effectively like statistical unknown would just be too much to me. I mean, of course, there's incredibly good work on it.
Right. That makes sense. In terms of what you just understand, though, about what it's doing in the brains of mice, even, would you assume that it is having some effect on human behavior, just based on what you know in terms of how it's infiltrating the brain and what you've seen in terms of the effect on mouse behavior?
Yeah. So there are some amazing similarities and amazing differences between the human and the mouse brain. So the fundamental thing that is true is that the neurons are mostly similar. There are some great papers out recently, like looking at this question in absolute detail. And you do find differences in, some like feature differences on the ends of human neurons and some genetic profile differences, but in general, they're the same. Like nature reuses the same parts over and over. This is true everywhere we look. And the first thing you might expect is, oh, it changes the mouse relationship to cat stimuli, to cat urine and like the presence of a cat. It switches in some of the animals this fear into an attraction. Maybe this is what predisposes cat ladies or humans that get it. You might take the analog and just map it one to one. But of course, we're not innately afraid of cats the same way that mice are. And so what we find in the mouse work is that there's an innate pathway that goes straight from the nose, skips all the fancy processing of the mammalian cortex, goes straight to like the hypothalamus, and then you get the behavior. But the human brain does not respond to cat urine or cat odors that same way. So the question is, if you have a bug, whatever that bug is, toxoplasma here, and I'm speaking colloquially, and the biggest difference, I think, and the thing that's hard to really truly appreciate is that when you have a chronic toxoplasma infection in a human, if even if it's subthreshold, it's not quite at toxoplasmosis levels, it's not medically relevant. That thing is sitting in the brain for decades, right? As opposed to a mouse whose lifespan is a couple years, maybe months or years. Having that same thing stay alive, we're just the ancillary host. We're the dead end. The parasite does not want to be in us. But I mean, we don't get eaten by cats, right? I mean, so we have no way of passing on the parasite. But what does it mean for this parasite to sit inside of a human neuron for decades? Is that neuron dead? Is that neuron forever altered? That's something that we will never be able to assess or study in a mouse because they just simply don't live long enough. And so there are fascinating questions about the human effects. And I really think, again, with the schizophrenia link, that is where you might imagine some sort of chronic immune response. So these parasites
Mm.
are, the immune system knows they're there. It's just not
Mm-hmm.
going to take out one of its own neurons
Right.
to get to it.
Right.
And so, this is really like wrapping yourself in the bear coat and, sitting in the bear cave. Like they can smell you. They know you're there, but they might not maul you just in
Yeah.
case you're
Launch
not
an
a bear.
attack.
And
Yeah.
so we know so little about the immune system.
Yeah.
We know so little about the brain. We are in
Yeah.
the Babylonian era of understanding how the central nervous system coordinates and works. And so I do think that it's quite plausible that if there are some molecular effects, if it's basically causing the neuron that it invades to be unhealthy in some redox or stress way, that accumulating this over decades could very well lead to chronic inflammation, changes in behavior, things like that.
Interesting. Is there a way to detect a toxoplasma infection of a neuron besides dissecting the actual brain? I mean, can it be detected through some kind of scan or fMRI, or is there any way for us to see what's happening apart from once someone passes away, actually dissecting and truly seeing it?
Most of the ways that people learn they have or have once had a toxoplasma infection is through a blood draw. It's just antibodies. You can do an antibody test, which can tell you had it at one point,
Right.
but there's no way of actually knowing whether or not a person has it in their central nervous system unless it grows so large, basically bursts out of its neuron and replicates and it can create what's called a ring enhancing lesion, which can be picked up on MR scans, which is how people with HIV, you could see often because their immune system is compromised. So when the toxoparasite breaks out, it just kind of replicates unfettered. And so, as far as I know, nobody's done postmortem for Toxoplasma. This actually came up in, so at Stanford, there's a law and sciences kind of working group where we would just meet for pizza and beer and give presentations and ask questions to each other. And one time I got asked, so the science isn't good enough to be introduced in a normal case. Like you can't just say someone has toxoplasma and link to all these studies or behavioral effects. But in the rare event of a capital punishment case, the evidentiary standard is lowered because they want to give a fair shot to anything. You know, someone's life is on the line. So they lower the standard. And someone asked me, like, if I were put on stand, what I would say. So let's say someone committed a murder. It was highly violent and they tested positive for toxoplasma. Could I say that the toxoplasma had any effect on that act? And not at all, because all we have is the blood draw and we have no idea what that means about whether or not there's active toxin in the brain when the person had the infection. If it's past 6 months, there's no way to distinguish from the antibodies alone.
Right.
So it's a really, really difficult. This is what makes that the science of the human, the toxoplasma infected human linked to all kinds of different behavioral traits so difficult, is that you don't actually have a record of where it is in the brain, which might plausibly give you just kind of some kind of connect the dots stepping stones to really like formulate a good hypothesis about why. We just don't have
how
that.
it's affecting someone. Yeah. I mean, even with something like SSRIs, I mean, the difference in how each one affects a single brain and how the same drug can affect different brains differently. I mean, we just do not know enough about the brain to be able to tease out those types of effects, I think.
There's this old saying, I think I got this from my PhD advisor, Robert Spolsky. He said, you know, a headache is not the absence of Advil. And in the same way, being depressed is not the absence of an SSRI. Behaving well towards cats or domesticating them is not the absence of toxoplasma. And what I find so interesting, though, is this kind of narrative that people often give over to these impingements on their free will and consciousness. So when people find out, I mean, you should see the emails we get.
Yeah.
Right? When people find out they have toxoplasma, they find out that they had it as a kid, they impart this narrative story and arc to, you know, was it me or was it the parasite?
Right.
And you find this commonly in, is it me or is it my cancer? Is it me or is it my childhood? There's all these narrative imprimatures that kind of come along for the ride when you find out you have something like this. And to me, that's the most interesting part. Like, how and why do people explain their own behavior? And they're often so wrong.
It's so fascinating to consider all the variables that are at play in determining our behavior, which all feel so much like our own.